Welcome to Unit 1 Techniques for CUET PG Life Science Paper Code SCQP17. This unit forms the base of molecular biology, biochemistry, biotechnology, and cell biology. This lecture is part of our CUET PG 2026 revision series on the BioBrainzz YouTube channel, where we revise concepts in a clear and exam-focused way. I am assuming you have studied these topics before. Here, we will connect ideas, recall key points, and think exactly like the examiner. As we go ahead, keep asking yourself, what can CUET PG ask from this slide? Stay focused, make short notes if needed, and let us revise. Unit 1 step by step. Today we will revise 8 major techniques. First, chromatography, bimolecule separation. Second, spectroscopy, interaction of light with molecules. Third, microscopy, visualizing tiny structures. Fourth, electrophoresis, separation in an electric field. 5. Centrifugation, separating by centrifugal force. 6. Blotting techniques, southern blotting, northern blotting, western blotting. 7. PCR, DNA amplification and its variants. 8. Radioisotope techniques, radioactive traces and detection. This is the complete roadmap for today's session. Why experimental techniques matter. These techniques are the backbone of biological research like diagnostics, genomics, proteomics, and modern medicine's depth on them. PCR is used in COVID testing, centrifugation separates blood components, spectroscopy and electrophoresis help identify proteins. In CUET PG, questions focus on 4 areas: principle, instrumentation, application, and comparisons. So, revise every technique from these 4 angles. Chromatography is a separation technique based on 2 phases. The stationary phase does not move and the mobile phase is the solvent. Different molecules interact differently with these phases, so they migrate at different speeds and get separated. Separation depends on polarity, size, charge, and binding affinity. In short, separates molecules based on how strongly they interact with the stationary, whereas mobile phase. Types of chromatography. First, paper chromatography, simple polarity based. Second, thin layer chromatography, TLC, faster and better resolution than paper. Third, column chromatography, used for purification. 4. Ion exchange chromatography, based on charge. 5. Gel filtration or size exclusion, based on size. Large molecules elute first. 6. Affinity chromatography, highest specificity. 7. HPLC or FPLC, advanced column systems. 8. Gas chromatography, volatile compounds only. Always connect each topic with its main principle. Key chromatography principles: ion exchange separates by charge, gel filtration separates by size, large molecules first, affinity uses specific ligand binding, partition chromatography depends on polarity. Applications include protein purification, drug analysis, metabolomics, and biomolecular separation. Spectroscopy studies how light interacts with matter. It is used for identification, quantification, and structural analysis. A spectrophotometer has a light source, monochromator, sample holder, and detector. CUET may ask you to label these parts. Types of spectroscopy: UV visible, quantification and Lambert law, fluorescent, very sensitive detection, IR, infrared, functional groups and bond vibrations, NMR, detailed molecular structure, mass spectrometry, mass to charge ratio. Each type answers a different biological question. Lambert law. A is equal to epsilon Cl. Absorbance depends on concentration and path length. Increased concentration means absorbance increases. Double path length means absorbance doubles. Used for estimating DNA and proteins. CUET often asks numericals from this formula. Microscopy. Structures invisible to the eye can be visualized using microscope. Resolution depends on wavelength. Shorter wavelength means better resolution. Light microscope limit is about 0.2 mm. Know the basic parts: eyepiece, objective, stage, light source, and focus knob. Types of microscopy: bright field, routine stained samples, phase contrast, live unstained cells, fluorescence, labeled molecules, confocal, optical sessions, and 3D images. TEM, internal ultrastructure, SEM, surface details, atomic force microscopy, nanometry scale surface imaging. These pairings are very important for exams. Electron versus light microscopy. Light microscopy allows living cell but has low resolution. Electron microscopy gives very high resolution but needs dead specimens and vacuum. Remember, light microscopy, live, low resolution. Next, electron microscopy, dead, high resolution, vacuum. Electrophoresis. Electrophoresis separates charged molecules in an electric field. Migration depends on size, charge, shape, and medium. DNA moves towards the positive electrode. Smaller fragments moves faster than gels. Types of electrophoresis: agarose, DNA by size, SDS phage, proteins by size 1D, native phage, size and charge, isoelectric focusing, PL, 2D electrophoresis, PL, then size, capillary electrophoresis, high resolution automated method. Applications of electrophoresis. Electrophoresis is used for DNA sizing, routine molecular weight, purity checks, genetic diagnostics, and forensics. Bands near wells are larger. Centrifugation. Centrifugation separates particles by size and density. Heavier particles form a pellet, lighter remaining in supernatant. RCF depends on radius and speed square. Blood separation is a classic example. Types of centrifugation. Differential centrifugation. Stepwise speeds. Crude fractions. Rate zonal. Size based and time dependent. Isotronic. Density based equilibrium. Analytical. Measurement only. Ultracentrifugation for very small particles only. Applications of centrifugation used for organelle isolation, virus purification, ribosome separation, and blood fractionation. Know the order of pellet formation with increasing speed. Blotting means transferring biomolecules from gel to membrane for detection. This involves blotting techniques like southern blotting, DNA, northern blotting, RNA, western blotting, protein, southern western blotting, DNA binding proteins, eastern blotting, post-translational modifications. Western blot steps, STS phage, transfer, blocking, primary antibody, secondary antibody, detection. Blocking prevents non-specific binding. Polymerase chain reaction, PCR, amplifies DNA in vitro and was invented by Carey Mullis. It requires template DNA, primers, DNTPs, Taq polymerase, and magnesium buffer. PCR steps, denaturation at 95 degrees Celsius, annealing at 50 to 65 degrees Celsius, extension at 72 degrees Celsius, repeated 25 to 35 cycles for exponential amplification. Types of PCR. RT-PCR, RNA to cDNA. qPCR, real-time quantification. Multiplex PCR, multiple targets. Nested PCR, high specificity. Touchdown PCR improves primer binding. Radioisotopes act as a tracers detected by autoradiography, scintillation counters, and Geiger counter. Applications of radioisotopes used for DNA replication studies, metabolic pathway tracing, receptor binding assays, diagnostic imaging, and pulse chase experiments. Revise these high-yield areas: principles of techniques, method comparisons, Beer-Lambert numericals, southern versus northern versus western, differential versus density gradient centrifugation, PCR variants. Our student support package is optional and linked in the description box. It includes Unit 1 notes, previous year questions MCQs, expected questions in 2026 CUET PG program, study schedules, and revision sets. If you want structured guidance for CUET PG 2026, you can explore this option or email us for specific queries. If these lectures are helping you, please subscribe to BioBrain channel for free CUET PG Life Science preparation. Thank you.